Welcome to the 𝘚𝘏𝘐𝘎𝘈 𝘚𝘏𝘐 product demonstration. This video reviews the method when using direct fecal specimens. These materials are included in each test kit. Refer to the PI for additional materials that may be required. First, add the diluent to a tube. Next, add 1 drop of conjugate. Transfer the fecal specimen to the tube. Close the tube and mix the specimen thoroughly, either by vortexing or by repeatedly inverting the tube. Using a new graduated transfer pipette, transfer 500 microliters of the diluted specimen to the small sample well on the 𝘚𝘏𝘐𝘕 device. Incubate for 15 minutes at room temperature. Add 300 microliters wash buffer to the large reaction window on the 𝘚𝘏𝘐𝘕 device. Add 2 drops of substrate to the large reaction window. Incubate for 10 minutes at room temperature. Read the results promptly at 10 minutes. Refer to the 𝘚𝘏𝘐𝘕 𝘘𝘜𝐐 package insert for detailed interpretation guidelines.